All right, everybody. We're going to go ahead and get started. Thank you so much for being here. I am so excited to see this auditorium almost full. My name is Cinnamon Moffett. I'm the research program manager here at Oregon State University's Hatfield Marine Science Center, and I will be your host this evening. Just a reminder, this event is a hybrid event, so welcome to those folks that we have online as well. Just a reminder for those folks, if you have any questions, Roseanne is in the room and she will be able to help you with any technical issues. So go ahead and use your chat and ask her any questions. Also, if you have any questions for our presenters, put them into the chat and we will work through those as we get to that place where we ask questions. For folks in the room, because it's a hybrid event, it does mean that you will need a mic to ask a question so that folks online can hear as well. And there's two of us that have these handheld mics. So just raise your hand when we get to the question and answer section and we'll run around and get your questions. So you get to watch me run up and down the stairs. It'll be fun. I also just wanted to let folks know that we are trying something a little bit new here tonight. And we have some technology that is something that you might just get off of a standard shelf. We have some GoPros that we're using, but that is a little bit of an experiment. So be patient with us. We're going to do the best we can to make sure you have a very good experience. But we're trying to do something this room isn't quite set up for. So we'll get there. I wanted to let folks know that for this evening, we're going to start with a short talk. We'll do a short intermission as we bring in our shark for dissection, and then we'll do the shark dissection component. So that's kind of the flow of the evening. I also wanted to make just a really quick announcement. We do these Science on Taps every month. They changed on the day. So if you are interested in learning more about them, you either can sign up for the listserv that's on the sign up sheet by the door, or you can go to HMSC's homepage, scroll to the bottom. There's a calendar of events there, and it'll list all the login information you need for any of our events. So our next one is going to be November 30th, when Lee Torres with the Gym Lab is going to be talking about gray whale research here off the Oregon coast. So we're excited for that one as well. But the reason you are all here is for our amazing presentation this evening from the Big Fish Lab. So I'm going to just do a really quick introduction and then I'll get them started with that. So I've got to put my glasses on. All right, so for this evening, we have Dr. Taylor Chapple, who leads the Big Fish Lab from Oregon State University's Hatfield Marine Science Center. He studies sharks and other large predators around the world, focusing on their movement, behavior, population dynamics. In Oregon, he is studying the sharks off our coast here, and so that's what we'll be learning about. He also has, and we have this evening, Alex, who is a postdoctoral fellow here with the Big Fish Lab. Her work is working on diet and habit of salmon sharks. So we're excited to have Alex. In addition to those two, we have other graduate students. We have Josh and Jessica here that will also be helping with the dissection component. So it is a full house from the Big Fish Lab. So again, we are excited they are here. Taylor, you have anything you wanted to add?
Cool. Thank you, Cinnamon. Thanks, folks, for being here. Yeah, I guess I was on the flyer, but I'm not actually going to do any of the talking. You all lucked out. So all the wonderful folks in the Big Fish Lab are going to do all the hard work for me. I'm going to sit over here, my son, and hang out and watch. But I did want to recognize them. You probably saw all of them wearing the Big Fish Lab gear. But if y'all could stand up just to make yourselves available. There you go. Y'all got a round of applause and you haven't even done anything yet. Nice work. Braden, where's Braden and Ethan? Are they not in here? Okay. All right. So Braden and Ethan, they'll come in a few minutes. Also, undergrads and recent grad volunteers that are working the lab. So just want to recognize them. One person that didn't get mentioned was Kyle Newton over in the corner. Kyle's also a research associate here. Braden and Ethan, thanks. So, tonight's just meant to be fun. It's just supposed to be an exploration of sharks. And so Alex is going to start us off with a little bit of background. And as Cinnamon said, we're going to do a brief intermission and then bring the animal in. So we'll do the dissection here. And then we ask that folks afterwards will go outside. As Cinnamon said, this space is not made for this. We got special permissions and a lot of convincing had to happen. So we're going to try to keep all of the blood and guts and gore up here. And then we'll probably take the animal outside if you all want to see and touch and smell. You might smell it from there anyway. Anyway, with that, Alex, take it away.
Thanks, Taylor. Hello, everyone. Can you hear me? This is so fancy. I'm not used to speaking with a mic. All right. Yeah, so my name is Alex McInturf. I am a postdoc. Cinnamon, am I standing in the right place? Okay. Oh, I'm gonna give this presentation here. Okay, cool. Before it gets really dirty. Hello. So I'm obviously not what you expect to see in this pool right now, but I'm going to talk to you for about 10 minutes about why we're doing what we're doing here in the Big Fish Lab, if this will work for me. Nice. Okay, so again, like Taylor said, we're a big team, so I'm just speaking on behalf of all of us today. I'm going to throw a lot of information at you, so if you have any particular questions about any of the pictures you see or the stories that I'm telling you, I encourage you to come talk to us afterwards. All right, this coastline should look familiar to you. It's very pretty, right? Have any of you seen sharks along the coastline here? I can't really see you all, so you got to yell. Okay, yeah, I'm sorry, I'm seeing some hands. Yeah, okay. Any idea what kinds of sharks you saw? Blue, salmon, nice. Hammerheads, Kyle, interesting. Okay, how many of you actually didn't know there were sharks in Oregon before tonight? Okay, yeah, so when I first started here with Taylor, I actually was a little bit confused about why I was coming to Oregon to study sharks. I had worked in the Bahamas and South Africa and California, so it seemed like a really odd place to find sharks. But in fact, they are big parts of the ecosystem here, which also includes a lot of other important fish, right? Like salmon. Now, to date, a lot of research has focused on how different animals in these ecosystems interact. And a lot of people are focused on predators like marine mammals. I know we have some marine mammal people in here, like whales and seals and sea lions and even birds. That person should look familiar to you. He just stood up to talk, except for he has a handlebar mustache in that picture. So there's a lot of work as well on what the people are doing here. Guess what's missing from this picture to date? Yeah, that's not a surprise, right? That's why you're here. That's why we're here. So yeah, to date, there really hasn't been any directed studies of what sharks are doing in the ecosystems here. How many are there? What are they doing here year round? Are they here for part of the year? And what are they eating, right? So that's what our lab is designed to do. Now there are approximately, I should say, 15 species of shark in Oregon, and they're really diverse, right? They range from 2 feet long to even 20 to 30 feet long. And if you want to learn more about them, I recommend that you visit our Shark ID page on our website, because I do not have time to give you fun facts about every single species today. And I'm just going to run through a few of the species that we are actively studying, but you should know that we are hoping to kind of continue to study these other species in the future. So as Cinnamon mentioned, I am currently working on a salmon shark project. You are going to see a little salmon shark here today, which I'm really excited about. We also have Jess Schulte, who's studying broad-nosed seven-gill sharks. Both Jess and I are really interested in what these sharks are eating in Oregon and where they're going. Part of our project is also in the Big Fish Lab is also on basking sharks. We study them both along the west coast of the US and in Ireland. That's a project that me and Taylor are working on. This is the world's second largest fish, and they eat plankton. So what you're seeing right now is the shark filter feeding plankton in the water. This one was in Ireland, but we are hoping to see them in Oregon. So if you see a big thing that looks like a sea monster and a white shark combined, please let us know. Probably a basking shark. There are white sharks here in Oregon, and that is not to say you should stay out of the water. You should probably stay out because it's really cold. But the white sharks are probably not going to interact with you very much. They are here, and so Josh Bowman and Taylor are working on tracking their movements and trying to figure out what they're doing here in particular. And we also have some really cool other studies going on in the lab. For example, Kyle is working on figuring out how sharks and their relatives respond to offshore marine renewable energy infrastructure, which is obviously a very noble pursuit. So we'll talk a little bit about that. This is way over my head. So if you want to learn more about that, I would talk to Kyle after this. And in addition to those projects, like Taylor said, we have a ton of other people with a lot of different specialties and interests at a variety of different career stages who are working with us. And we all help each other out, and it's been a lot of fun to work with them. So anybody who's wearing a big Fish Lab sweatshirt or t-shirt, you should talk to today because they definitely have a cool shark story to share. Right, so it's really hard to study sharks. I don't know if that's like, news to you all or not. It's really hard to study animals that we cannot consistently observe, right? Unlike marine mammals, they do not have to surface in order to breathe, and we cannot breathe underwater. So we cannot watch them for a huge part of the time that they're doing these critical behaviors in Oregon. So we study sharks in a few different ways. And the first is, of course, we go out and try to see them in the field, in the wild, in their natural habitat. And we have to get a little bit creative, especially when the shark is bigger than your boat. So for example, this is a drone shot of a basking shark. That shark was about 7 meters long, so that's like 21 feet. And we can use drones to look at whether sharks are in an area if they're at the surface. And we can even use them to try to see how big those sharks are relative to the size of our vessels. We also have a variety of different tags. Have you all heard of tags? Yeah, we had a few out there as well, if you got to see them. This is really just a general term. It's a device that measures movement or behavior of different animals, right? We can't just stick a GPS tracker on a shark, right? That doesn't work very well underwater typically. People will try. So we have a few different tags that allow us to ask a few different questions. An acoustic tag, for example, is kind of like a Bluetooth. If you were out there, you saw an acoustic tag, right? That was the really annoying pinging sound that was happening at our table. So those are really useful if we want to know if a shark is in an area. We can lower a device called a receiver from the boat, or it can be moored on the sea floor. And we can actually listen for those pings from tagged sharks. And we can say, hey, shark 45243 was in the area. And we know that we tagged that animal here 5 years ago. So that shark's probably coming back. So that's really good for looking at whether the sharks are around and even their short-term movement patterns. We also do have satellite tags. We had those out there as well. These are more similar to those GPS trackers, right? They give us information on long-term movements of sharks. And then we have camera tags, which are super cool. They're also like animal Fitbits, right? So they're like the things you have in your phone or on your watch that track your steps. We have those in sharks. They measure the shark's body movement, and that tells us a little bit more about what behaviors those sharks are exhibiting. And then we have the camera to back that up, which I'll show you in just a second. So we decide to choose, I have to leave my pool, hold on. We choose our tags based on the questions we're interested in asking, but how do you put a tag on? Oh no. This is footage of a white shark being tagged off the coast of California. The shark will approach the boat here, and then you'll see that pole dart, it looks like we're harpooning them. They are okay. And they will behave pretty normally after we poke them with the tag, and then the tag will be placed just under the skin on top of the shark. So we'll do something like that for white sharks or for basking sharks. And then the tags will look a little bit like that. That shark here has an acoustic tag and a satellite tag on it. And for something like Jess's project, though, we can actually bring the shark alongside the boat. It is not bigger than our boat, so that really helps. And we can do what's called a workup alongside the boat. So here you're seeing a bunch of pictures of Jess and Taylor working up a shark, getting information on its size and its sex, and also putting tags on the sharks that way. And again, we always try to make sure that the sharks are behaving pretty normally after we release them. So here's an awesome video of a successful release of a seven-gill. Now those camera tags are really cool. So this was a camera tagged salmon shark off the coast of Alaska. We also have satellite tags on these animals. So in addition to knowing those long-term movement patterns from the satellite tag, these camera tags are really cool because they can tell us what the animals are doing in a moment by moment basis, right? What are they doing when they're on the coast of Alaska? This one is just checking out the coastline as it turns out. So those camera tags are very useful for understanding what the animal is doing and seeing from a shark's eye view. And this is another one I have to leave this for. And then we can start to look at things like where the animals are going on Earth. So this is, again, a white shark series of tracks from satellite tags. These are coming from California and from Mexico. And you can see there's no way we would have known those sharks were ending up in the middle of the Pacific Ocean without the use of these tags. And now researchers can start to understand why they're going to these locations. Now, I also do a lot of stuff in the lab. This is a little bit gory. If it's too gory for you, you probably shouldn't stay for the dissection. So dissections are super important for us. Part of my work is trying to figure out what these sharks are eating. So we do dissections a lot and just rifle through their stomach contents all the time, which is really fun. We also have 2 experimental projects going on. As you probably saw at our table, we have a bat ray project, which Josh is working on, and I'm not going to go into detail on that, but you should talk to him after this. And Kyle is also working on trying to figure out experimentally what happens if he has skates, which are cousins of sharks. What happens if he changes the geomagnetic field in a given tank? How do they respond? So Kyle is working to manipulate the environment that these animals are in to see how they might respond if we have that infrastructure out in the water also. And finally, part of the reason we're here is we really enjoy engaging with the community. And in fact, we really have to. There haven't been a lot of studies published, again, on sharks in Oregon, basically none. So we are trying to learn from people who are in, on, around the water, who know the water in the area much better than we do, to try to figure out some baseline knowledge about a lot of the species that we're studying here. So we do have a reportings page on our website. Those little cards that we were handing out also have those links. And we are asking people to tell us when they're seeing sharks, what species they're seeing if possible. And that will allow us to try to target our research and tagging efforts. There's a dead salmon shark in the middle behind me. You will see that again very soon. We are really interested in strandings as well. So if you see a dead animal, certainly it's unfortunate, but it's useful for us to try to understand what that animal was doing in Oregon waters. If you see a live shark on the beach, also, we can help you try to figure out what to do with that. And finally, if you like learning about sharks, we love talking to people about sharks, like I said. So you should visit our outreach page. We also have tools for educators, and we have the most recent up-to-date stories covering our research so you can learn a little bit more about what we're doing and stay up to date yourselves. And now it's dissection time. So thank you so much, Josh.
Awesome. Thanks, Alex. So we are going to take just a couple minutes to go outside and find a shark and wrangle it and bring it inside. So give us 2 or 3 minutes. If anybody needs to go have another slug of their beer outside, do that. And then we'll meet you back here in about 3 minutes.
All right, everybody. I think we are ready to go. So go ahead, lights off. Josh, are you green? Green light.
Are we ready? Hello? Okay, let me guys introduce you to the star of the show tonight, which is this salmon shark. I realize it might be a little bit odd that I'm up here talking about sharks with a bandaged finger that is not from a shark attack. My own mistake. So what we have today, let me come around to you, is so this salmon shark, we got this animal from beach stranding, which is fairly common for salmon sharks or sharks of this species. And this is a juvenile. So it's about, within the first year of its life. And I'll start by explaining that these are warm-blooded fish. So they're regionally endothermic. And that's different than most sharks, which have their internal body temperature or their whole body temperature is regulated by the temperature of the surrounding environment. And so what happens with these juveniles is they get subject to a thermal shock, which is from the cold water. And then that causes them to not be able to regulate as they normally would, because otherwise they're specialized and designed to perform very, very well in cold water. That's part of what that warm-blooded strategy is for. But these juveniles aren't quite as big and able to deal with that thermal shock. And so what happens is they get washed ashore, stranded, and then die. And folks may have come across them and mistaken them for a baby white shark because they do look rather similar. They're from the same family of warm-blooded sharks, so they are related to white sharks. But white sharks come out at about 5 feet long, and this is much smaller. And there's some other notable differences between salmon sharks and white sharks as well. One of those being the back of the tail here is called, so this is the caudal fin or the tail fin, and this joint here is the caudal peduncle. And so it has these ridges that go alongside it of this animal that's otherwise sort of built like a torpedo. So really designed for speed and dynamic performance in the water. And these ridge lines on salmon sharks, if you are able to see it, you might have to get really closely. Does it? Okay. Yeah. Anyway, there's a really small secondary caudal keel, and so this is the main caudal keel is what you see with that ridge line that goes along the tail. And part of what that does is provide extra structure for the animal, so it can utilize all of the larger muscles up here, and the upper portion of the animal stays rather rigid, while this lower portion with the tail does all the movement and is what propels and pushes the animal through the water. Are we back with the camera quite yet? Or okay, yeah, no worries. Some other big things that you may be able to see from the main camera or in the audience. But did we see Taylor's face?
Yeah.
Awesome.
This is my first time on Zoom.
For folks online, we're having a little trouble with the camera. We're getting it set back up again. Hang in there with us.
Cool. Okay. So what I was just explaining is how the animal moves, how it propels itself through the water. So most of that thrust is generated by the tail, which is the upper portion of the animal, which where the dorsal fin and the pectoral fins are. So this being the dorsal fin and these fins on the sides being the pectoral fins. That part stays rather rigid, while the tail is what moves back and forth, kind of like a tuna, to move it through the water. And so they are designed in a similar way that other fast-moving, high-performing fish are, and they're very, very built for speed to chase down and overcome fast-moving, elusive prey. And another strategy they have for that, so mostly when we think of sharks, we think of their teeth, and so that's another tool that they have to keep pinned down the elusive prey when they are able to get it. Can you see this? Cool. So on a salmon shark, and also unlike a white shark, which has very broad triangular serrated teeth, these salmon sharks have much more narrow, thin, and pointy teeth, which is more ideal for spearing and holding down holding down your prey once you get it. And whereas I'll use the comparison as a white shark is more for cutting through flesh of their prey preference, which is marine mammals. Also on the head are their sensatory organs. So sharks have some similar sensory capabilities as us. They also have some sensory capabilities that we don't have. The ones that are similar are going to be eyes, which you can see here. So the group of sharks that salmon sharks belong to, they don't have nictitating membranes, which is what a lot of sharks do. It's a third eyelid that can close over and protect the eye while feeding. Instead, the eyes will roll back in their head when they go to bite something. They also have nose pores, or what we call nares. And those are similarly used for the sense of smell. Can you freeze the camera again? Yeah. Okay. So it's similar to us. Their nares are used for their sense of smell, but unlike us, they don't breathe through their noses. So we can use our noses to do both. We can smell and breathe. For sharks, it's just for smelling. There's no breathing component associated with it. And so they actually have two pores on each nostril, an in-current nare and an ex-current nare. And then these sort of jelly kind of globs that you see along the face of the shark, this is where they start to have different sensory capabilities than what we're used to. These are the pores, the ampullae of Lorenzini, which is what they use to detect like electrical changes in electrical current, in electrical fields. And so these are just the pores. These actually connect to long canals that go back to the ampullae of Lorenzini that are filled with a conductive jelly fluid.
We're back.
We're back. Sweet. So yeah, we can flip the shark over and get ready to look at the inside. So we'll talk a little bit about shark skin, which is what we're about to cut through. So shark skin is covered in these little tiny scales that aren't really scales, but we call them dermal denticles. And so these, one, they protect the shark. They're very tough. But two, they also reduce friction of the shark as it moves through the water. It prevents these development of like turbulent eddies and water flow that create drag when the shark moves through the water. So without those, it's able to move through the water much more efficiently. And so this technology was used in swimsuit design in the Olympics, and it actually got pulled out. It was banned because it worked so well that people were just smashing world records, and it didn't seem very fair. So that's how successful that strategy is for their movement. So as Alex is cutting open this animal, the first thing that we might notice is this really smooth, big organ. That's the shark's liver. And the liver for a shark is especially important because unlike bony fishes, they don't have air bladders or swim bladders. So a way for them to, like what a bony fish might do to control its buoyancy in the water is fill that air bladder with air and deflate it as it wants to descend, fill it with air when it wants to ascend. Sharks don't have this. And so instead what they use for buoyancy and lift is these much larger livers. And those livers are filled with fats, and those are positively buoyant, which help provide lift for the animal. Another way they generate lift is their cartilaginous skeleton, which is another like unique feature to sharks and their relatives. They don't have any bones in their skeleton. It's all made of cartilage, which is less dense and lighter than bone. So Alex is going to remove this liver. One of the reasons that sharks were hunted historically was for these livers. Being so full of fats, it makes it a really good and a really convenient place to store vitamins, many of which are fat soluble, like vitamin A, which before we could synthesize was a large reason why soup fin sharks or school sharks were harvested along the coastlines here, was for their large livers full of vitamin A, and that was one of the ways that we sourced it. So you can see how big these livers are relative to their size. They can be up to 30% of the animal's total mass and take up 90% of the room in the body cavity itself. So there's two lobes, one liver. It looks like two livers. That's actually all part of the same organ. And as soon as we get the liver out, this would be a good time to talk about how we determine sex in sharks. So this shark is a male, and we can see that by the presence of these two claspers. So only males have these two claspers. They both have cloacas, which is what the opening here is. That's how the shark expels its waste. But the claspers are another interesting thing that we can tell with these is if the animal is mature or not. And I know I said earlier this is a juvenile shark, but one of the things that as a shark gets older, these claspers, which are analogous to penises, they will get more rigid and stiff. When they're immature, they're like really bendy. You can see that there isn't a whole lot of a whole lot of structure in its claspers. And what's harder to see that I'll explain on the bottom of the shark is in male sharks, they have these features called siphon sacs, which are these very like shallow sacs along the underside of the shark that are used to pump seawater in. And that's part of how the animal can ejaculate its sperm is to fill those siphon sacs with water and then use it to flush out sperm through those ducts that go along the body. They don't have another way to ejaculate their sperm for copulation. We're getting some
Going deep
dive into sharks. So right under the liver and in the body cavity here, this is really we got this animal really early on. Sometimes we get animals they've been scavenged on before. This isn't the case. This is actually quite a exceptional condition to be able to do this kind of demonstration for y'all. So starting with the heart, which is up here, just underneath just like up against the jaw, up in that cavity. The heart on sharks so it has one ventricle and one atrium, but it's actually a 4-chambered heart, and it has 2 other smaller chambers that they function as more of an inline system. So blood will flow one direction through the heart, going from the gills back to the body, and then it returns to the gills again. So it doesn't go through the heart twice like it would in humans. And then we have the digestive tract. So we'll pull the heart out, put it here, heart nipple tray. And so the digestive tract is just under the livers. And so what happens when the shark eats its food, it goes into its esophagus through its sphincter, which is the muscle that keeps food from flowing back up through the esophagus into the stomach. And so normally what we'd like to do is open up the stomachs and show you guys what is inside it. But so part of Dr. Alex McInturf's work is to do the stomach content analysis for the foraging ecology for these animals to try and better understand what they eat while they're in Oregon and what their role is here. So we'll zip tie off those openings. So we want to be able to go through that in a controlled setting in the laboratory. So we can show you the outside of the stomach, but we want to save the inside for our analysis.
Also, it smells really bad when I cut it off.
Yeah.
Welcome.
Particularly as the stomach moves along. So this is the stomach here. As that travels to the duodenum, the intestine is, so in humans, we have this really long, curvy intestine that does all these loops and stuff inside. Sharks have, and that's to increase like surface area to help us uptake as much nutrients out of our food as possible. Sharks have a similar strategy, just instead of being very long and bundled up, they have a ribbon-like structure that spirals through the intestine itself. And you can kind of see the edges of that, which are these lines here. Those are the edges of that ribbon that spirals through the intestine. So food will move along that like spiral circuit. And so it actually it's not just a straight pipe that goes out to the rectal gland and anus.
Can I stop for 1 second while Alex is pulling that stomach out? I do want to remind folks that Ethan and Braden are lining up these awesome trays that we're putting different parts on. And then we're going to take those outside. So if y'all want to get a really good look at a spiral valve, that's your chance. So even if you can't see a lot of the definition now as it's all coming out, we will put it outside so y'all can like a scratch and sniff experience
One on
outside
the-
for anybody that
one on the
wants.
upper end of the stomach. Okay. Sweet.
Well, this is also a good time if anybody has questions while we're pulling these parts out about any part of this. This would be a great time to ask them.
Does the liver taste better to us than the meat?
That is a question I would not be able to answer. I have not eaten a salmon, shark meat or liver. But I would imagine it's up to personal preference.
Cinnamon, do we have toothpicks outside that we can?
Yeah. Do a
That might be pushing the boundaries of what we're allowed to do with our public today. Any other questions online? Okay, hang on just a second.
Out of all the sharks that you're studying, what are most common in our waters?
That's a great question. It depends on where you are at. So in the offshore environment, we'll have a different collection of shark species than the near shore coastal environment. So if you're offshore, one of the species that you might expect to see are blue sharks. So there's blue sharks are fairly abundant in the offshore environment of Oregon. If you're in the inshore habitat, like say within a large estuary or a bay, then you might see a lot more seven gills that are in those environments. So it largely depends. The animals that we see the most of are going to be blue sharks and seven gills. And so yeah, hopefully that answers your question.
All right, are there any questions online? All right, for folks online, just go ahead and put those questions into the chat box and we will work through them. All right, I have a question that's about halfway up, so here we go.
How do sharks swim if they don't have any bones in them?
Sorry, could you repeat that question?
How
I
do
didn't
they
quite get it.
swim if they have no bones?
How they swim if they have no bones? So instead of bones, they have cartilaginous structures that are shaped similarly to bones would. So they still have a skeleton. It's just made out of a slightly different material than a calcified bone would be. So they would use that skeleton in the same way they would use calcified bone. Just instead of being calcified bone, it's made out of cartilage, like your nose or your ears. Thank you.
We still going with questions? All right, we got one more right here.
Are there any shark body parts that are similar to a squid's?
Are there any shark body parts that are similar to a squid's? One that jumps out at me right away would be eyes. So sharks and squids both have eyes. They have mouths, although they would look much different, being a squid having a beak and sharks having those toothed jaws. But no tentacles on a shark. So they definitely have their differences as well.
All right, go ahead.
What is the fat made out of?
Which fat?
The fat on the outside of the skin.
So he wants to know about that layer between the skin and the muscle. Josh, I told Josh that he's gonna have to answer any question that came out. So even the ringers.
Okay, sharks have very little fat on. Was there another question? Oh,
The
sorry.
camera is frozen again.
Okay, sharks have very little fat on their bodies. Most of the material between the skin and the muscle is a network of fibers. They call it, and what we call it is an exotendon, but it works similarly as like a network of fibers that provides strength when you pull on it, and it is more flexible when it relaxes. So they don't have a lot of fat between the skin and the muscle. A lot of their fat is stored in those livers. So that's part of the reason why many more commonly, sharks are cold-blooded, is so they don't have that fat to insulate their bodies from the surrounding water environment. And so the heat dissipates from them immediately.
All right, we're going to hold questions and we're going to get back to the dissection. We'll come back to questions in a second. If you are ready, Josh.
So right beneath here, these kind of soupy structures are the testes in a male shark, followed by the kidneys, which we move these out of the way. The kidneys they're a little bit hidden, but we can open up that tissue. So what we're used to in a kidney is kind of like that bean-shaped organ. For sharks and fish, that organ looks quite different. It goes along their entire spine, and in their body cavity is all these two long kidney tracts, if you will. No, that's perfect. Yeah, so commonly this is referred to as the bloodline in fish because it is red color and chunky, and it has the appearance of being blood. It's actually their kidney organ, which helps them like process urea, which sharks would normally expel, or animals would normally expel. Sharks reuptake and keep urea in their bodies. So as we work closer and closer towards the spine of this animal, is a good time to talk about how that regional endothermy process works, and how we can just talk about it generally now, and then we'll do a cross section and show some of the things. Yeah, good. So these animals, as we mentioned, are somewhat unique in that they can hold on to the body heat that's generated by their muscle contractions. So part of what happens when you move your body is those muscles generate heat. In a exothermic fish or a cold-blooded fish, that the blood that returns the veins that return the heated blood back to the gills go along the outside of the animal near the skin. And so because they don't have a lot of insulation, that heat is lost rapidly to the environment. The other big location where heat is lost is at the gills. So about 30% of body heat is lost at the gills. And so sharks have to breathe, and they have to breathe through their gills, so they can't really get around that part. But they can get around the part where their vein structure brings their warmed blood along the surface of their skin. And they have a network of veins and arteries that are up against the vertebrae of the animal. So they're much more insulated by the muscle itself and go along the vertebral column. And so that helps them maintain the heat that's generated, that their muscles that do a lot of the steady state locomotion, or the red tissue. In vertebrates, we have red tissue muscle and white tissue muscle. And the red tissue muscle is what is used for the constant motion. So like the muscles you use in your legs for walking. We know this in similar like chickens that have white meat and dark meat. And so that dark meat is the muscles that do a lot of the steady, constant work. In warm-blooded sharks, that muscle also goes along the vertebral column. So that muscle that's constantly generating heat is located inside, up against the, insulated by the rest of the animal's tissue. And so that's how they maintain and capture the heat generated instead of losing it to the environment. And so we can show what that muscle looks like, taking a cross section of the animal. We'll actually get to see where those muscle groups are and what it looks like. So I'll just cut this shark in half. And so we can see that all the muscle tissue around this cross section is all white, except for these little patches here that are that dark tissue. And so that's what the animal uses to in a cold water in a exothermic or cold-blooded fish, that red tissue would be located along the outside of the skin here, not insulated up close against the vertebral column there. And so yeah, if we have questions from folks, that would be a great time. That is what we had planned for y'all.
All right, so we're going to open it up. And again, for the folks online, go ahead and put your questions in the chat. And I will check on you and see if there are any questions for folks in the room. Just raise your hand. And you had a question?
I wanted to know if all species of sharks have teeth that they rotate out to drop.
Well, sorry, could you repeat that?
It's a little hard for them to hear the questions because they're behind our mics and our speakers. So do all sharks have teeth that fall out and rotate up into place?
Oh, yeah, that's a good point. So yeah, all sharks have this, they are constantly regenerating teeth so that as that new row comes in, the old row falls out. And they also do that with the dermal denticles. So if they were to lose a patch of those denticles covering their skin, those would replace themselves, and they do replace themselves continually like their teeth.
All right, we got another question up here.
What are some primary reasons for strandings on the coast of Oregon?
So one of the primary reasons for strandings of these salmon sharks is that thermal shock, where the smaller animals are not quite as able to deal with their cold environment as the adults, and so they lose their function because they essentially lose too much body heat to maintain their normal functionality, and so they get stranded because of that. But other sharks get stranded. That can be from a number of reasons. Either the animal gets sick or has depleted its energy from a fishery interaction, or is in some other condition where it's not otherwise able to regulate its position in the water as it normally would.
All right, any questions online? Not yet. Okay, I got a question right here.
What are the most common causes, like human related, for shark deaths?
Human related for shark deaths. I don't know that I know the specific fishery interaction, but it's largely from bycatch. So sharks that get caught incidentally from another fishery that's targeting a different type, or from directed fishing where they're fished for their fins and meat, liver and teeth and those kinds of things. It would vary depending on location and where you were in the world. In some places it may be more so due to the bycatch, and in others it may be more from the directed fishing of sharks.
Is there anything to say that urea attracts sharks, like from other fish, or maybe even like in the wetsuit of a surfer or something like that?
Is it safe to pee in your wetsuit?
We get this a lot.
Yeah, that's not the first time we've been asked that. I will leave that up to the individual, like the liver and the muscle thing. They would be able to sense that you peed in the environment if they were nearby, so they'd be aware of it. Whether they're attracted to that or not, I don't know that there's enough information to suggest one way or the other. In other words, that hasn't been very well studied.
I'll add on to that just a little bit, that that they know that you're there a lot earlier than smelling your pee. So yeah, they're definitely going to be, they know that you're in the area, and that they're making a decision, and it's not going to matter about whether they can smell your urea or not. What's that?
The question is that vibration that they know that you're there?
There's a lot of different ways that they know that you're there. So I mean, Josh went over some of the different sensory systems that they have. Sorry, I'm like sitting down taking these questions. But there could be a number of different ways that whether it's the vibrations, whether it is it's actually the sound propagation. It could be a general smell. I mean, they can smell the neoprene and all the other chemicals that are coming off you as well. So and I think I tell this to everybody that if you've ever been in the ocean, there's a good chance that you've had a shark nearby you. So they know we're there a lot more often than we know they're there.
Okay, I'm just gonna let folks know in the room, we're gonna have more questions than we have time for, but you can ask folks questions at the end if you are here locally. So hang in there. And if you've already asked a question, you probably aren't gonna get another turn at the mic. So just hang in there.
What proportion of time do sharks spend at the surface? And have you ever seen any sharks with propeller scars from boats?
So the first part of that, time at the surface, that will vary by species. Typically, what it's typical in a lot of shark species is this oscillatory up and down motion that they do throughout the day and night. And for like boat strikes and propeller wounds, it's not always obvious to tell that an injury you see is from a boat strike, but we have seen sharks that appear to have been struck by boats. Yeah, go for it. Yes. Run it.
That much. Okay. I'm not going to touch this. So for the basking sharks, that's actually a really common problem in Ireland in particular. So Taylor and I have seen some pretty gnarly scars on the dorsal fins of basking sharks, but that is a trait that's pretty unique to that species as well. That's why their name is basking sharks, right? They like to bask in the sun. So it definitely depends on the species, but we see that pretty frequently over there.
All right, got another question right here.
It was super cool to see all the satellite tags and how the sharks spread out throughout the Pacific, and then they seem to cluster around the South Pacific. What are they doing out there?
So what are sharks doing in the South Pacific?
On one of Alex's slides, when we were looking at the tagging data.
Okay.
Yeah.
Yeah. So that's a spot. It's in the middle of the Pacific. It's sort of halfway between Baja, Mexico, and the Hawaiian Islands, and it's a spot that we call the White Shark Cafe. And it's because we have two sort of competing hypotheses about what they're doing out there. And so we came up, the only reasons that you'd leave this really rich coastal habitat is either for another rich resource, so there's something else to eat, or the opportunity to mate. And so currently the data are pointing to that may be the spot that they're heading out to mate. So we think that they may be going offshore to copulate, and then they come back. And why the cafe is the specific spot for it is still a bit up for debate.
All right, any questions online? No questions. We got a shy audience online. Okay, questions in the room are definitely aplenty.
So you mentioned that they go up and down cyclically throughout the day. Is that a diurnal effect based on light and dark? And if so, is the diurnal effect on the prey causal or uncertain of the behavior of the sharks?
Yeah, go for it. I'll take a stab at that first. So there's two parts. Part of how they move through the water is sort of they'll propel themselves up and then kind of glide down and then propel themselves up, so they're slightly negatively buoyant. And that's an energy conservation strategy. And then if Taylor, you want to get to the prey part of that with the diurnal effects.
Yeah. So as Josh said, there's part of it, which is there's multiple parts of it. One is that transiting. They do the same thing birds do, as Josh was sort of just explaining, is that because they're negatively buoyant, they'll swim to the surface and then they'll glide down and swim to the surface. Same way a bird does as you see them transiting through the air. But they also will follow prey, and some species have that sort of diurnal behavior where basically prey from down deep at night come up to the surface to eat the plankton that's on the surface. And then during the day, because there's nowhere to refuge, there isn't a bunch of trees to hide in, they have to head down deep. And so we see a lot of sharks that will follow that sort of up down motion. There's also animals that will, like hammerheads, that will dive down into the deep water, into basically the low oxygen, the low light levels where animals are hiding out. They'll go down there during the day as well. So it varies a little bit depending on the species.
All right, so believe it or not, we have spent an hour together. And so we are out of time for what we're going to do here. in the formal presentation, but we are going to stick around to be able to answer questions. What I ask, however, for those folks that are in the room, that you don't come up here, that you meet us outside. So we're going to do a little transition and we're going to take the animal and the parts out into the courtyard so that we can look at them out there. For everybody online, I just want to say thank you so much for being a part of this conversation. It is wonderful that we're able to have people in the room, and wherever you happen to be around the world, we're happy that you're here. And again, remember, if you are interested in these events, you can check out the calendar, and we're going to do another one on November 30th. So if all of you can thank our speakers, that would be amazing. All right, we'll see you next time. And if you want to hang out, come check us out outside. Okay, for everyone online, we're going to end the presentation. Thank you very much for being here.